Hi, my name is Sarah Jennings. Thank you for watching my talk, which is titled Smells Like Home, Bird-Scented Nests as a Mechanism for Olfactory Homing in a Burrow Nesting Seabird. Many animals make trips to and from a familiar site. This type of navigation is called homing, and chemical information frequently informs this behavior. Two categories of chemicals are recognized as aiding in this process. Organisms can use environmental chemicals associated with a specific place. Homing is also often facilitated by chemicals deposited in the environment by an individual or its conspecifics. I study the Leach's storm-petrel, which is an excellent system to investigate olfactory homing. The species is nocturnally active at the breeding colony, where they nest in an underground burrow. They use their sense of smell to locate both the breeding island and their burrow within the colony. Moreover, both adults and chicks can use odors to identify their nest. And finally, these birds have a strong body odor that may scent their burrow. In reality, homing is a multi-step process, which in storm-petrels involves locating the island where they breed, navigating through the colony, and finally distinguishing their home burrow from neighboring burrows. We predicted that there were possible roles for both environmental chemicals and storm-petrel-produced chemicals in facilitating this process. We tested two hypotheses. First, that Leach's storm-petrels use environmental odors for navigating to and moving around the colony. This hypothesis generated two predictions: that different geographic areas within the colony are associated with a specific chemical profile, and secondly, that samples from different landscape features are chemically distinct. Our second hypothesis stated that Leach's storm-petrels use self-deposited chemicals to identify their burrow, which had three associated predictions: that storm-petrel chemicals are elevated inside occupied burrows, that each burrow contains a unique blend of storm-petrel-derived chemicals, and finally, the types and quantities of storm-petrel-produced chemicals in the burrow match those found on the plumage of the occupants. To investigate these ideas, we examined the odor profiles of the colony landscape and its storm-petrel occupants. We gathered soil from two locations, from inside petrel burrows, and also from the colony floor immediately outside the burrow, which I will refer to as the background for the rest of this presentation. We sampled 44 background and 44 burrow locations across the colony, collecting 3 subsamples at each location. Overall, our data set contained 30 burrows that were occupied by storm-petrels and 14 that were unoccupied during this particular breeding season. We also removed birds from the burrows and collected feathers to study their odor profiles. We extracted the odor compounds emitted from the samples and analyzed the captured chemicals using gas-chromatography mass spectrometry. This allowed us to identify and measure the odor compounds associated with each sample. The data associated with each sample is highly complex, with many compounds in differing amounts. So I'm going to rely on ordination plots to visualize the patterns in the data. In this particular plot, the 2 colors and shapes represent 3 separate areas in our study colony. We found that the odor profiles of soil samples differed between these 3 locations. A number of plant-derived chemicals, which many birds are actually able to smell, were responsible for these site differences. So if storm-petrels can learn these place-specific odors, then they may be able to use them to facilitate homing. We also examined whether the odor profiles of the soil differed based on where in the landscape each sample was collected. We found that soil samples from occupied burrows where birds were actively breeding had different odor profiles than empty burrows and the background samples collected from the colony floor. We identified 7 chemicals that were on average 8 times more abundant inside occupied burrows than in either the background or the unoccupied burrow samples. Interestingly, these compounds were all major constituents of storm-petrel plumage odor, suggesting that the avian occupants of the burrow were the main source of these chemicals. We tested whether each occupied burrow contained a unique blend of these 7 storm-petrel chemicals. In this plot, each color and shape represents a set of 3 subsamples from an occupied burrow. You can see that the 3 subsamples cluster together, indicating that each burrow is chemically distinct. This result provided support for our prediction that olfactory nest recognition is facilitated by bird chemicals deposited inside the nest. However, as I mentioned earlier, we also collected feather samples from storm-petrels to analyze their odor, and so we wanted to take this analysis a step further. We examined how the scent of the burrow compares to the scent of the storm petrel that occupies it, to determine whether we could detect overlap between the odors of the burrows and birds. To do this analysis, we used samples from 19 burrows and 19 storm petrels, where the two sample types were collected at the same time, meaning we gathered the soil and then immediately reached into the burrow and took a sample from the bird inside. We specifically tested whether the blend of 7 storm petrel chemicals that were elevated inside the burrow matched with the blend of these chemicals detected on the storm petrel feathers. Here you're looking at 2 plots showing the chemical profiles of burrow samples on the left and storm petrel samples on the right. The colors and symbols match such that the burrow and the corresponding storm petrel use the same combination. If you look back and forth between the 2 plots, you can hopefully see that the burrow sample occupies a similar location in the plot as the corresponding occupant sample. Here are a few examples to help you see this pattern. We used a Procrustes analysis to determine the degree of overlap between the 2 chemical data sets, which calculates a goodness of fit score and a correlation between the 2 ordinations you can see here. We found significant overlap between the burrows and birds, which is to say we had evidence that the burrows are not only scented like storm petrels, but they actually reflect the scent of their specific occupant. We also compared the chemical profiles of 19 mated pairs, as there are 2 birds that could be impacting the compounds deposited inside the nest. We found that pairs were highly similar for the 7 storm petrel compounds that were elevated inside the burrows. In this figure, members of a pair are represented using the same color and symbol, and the 2 birds from the same location cluster together. This finding indicated that the storm petrel chemicals inside the burrow seem to reflect the identity of the pair and not just the individual who most recently entered the nest. In summary, we found support for both of our hypotheses. We discovered that the colony contains gradients of environmental chemicals and burrows that are scented like storm petrels, both of which likely aid olfactory homing in this species. I've also presented results that support the transfer of chemicals from storm petrel plumage onto their burrows, causing each nest to be scented like its occupants. Thank you so much for watching. Please feel free to contact me with any questions or to discuss these findings.